Now I humbly request our faculty, Dr. P. Sendil Kumar, to take the session chair now. I welcome you, sir.
Good afternoon, all of you. Now, Arunachalam Ramaiah, Professor Arunachalam Ramaiah is going to deliver his lecture. Before getting into the session, I would like to introduce the chief guest. It is my pleasure to introduce Professor Arunachalam Ramaiah, who is the group leader of Bioinformatician at City of Milwaukee Health Department, USA. He has obtained his Ph.D. in Bioinformatics from MS University and jointly from Abo Academy University, Finland. He has worked more than 12 years in India in various prestigious institutions like Tata Institute for Genetics and Society Bangalore and IISc Bangalore as a scientist. He got various awards from around the world, and few of them are prestigious ISID New Investigator Award from ISID USA, and he is the Fellow of Royal Society for Public Health and a member of Royal Society for Biology UK. He is also a fellow of Center for International Mobility, Finland. His research involved in genomic epidemiology of SARS-CoV-2 and its transmission patterns, and also his research involves in studying the RNA viruses like Zika virus, influenza virus, and SARS-2. He is the editor of various reputed journals like PLOS ONE and Frontiers in Microbiology and Frontiers in Ecology. He got more than 25 awards around the world, and he published very good papers in reputed journals like Genomics, Cell Reports, Journal of Immunology, Nature Communication, and PLOS Biology, and MedRev and BioRev. Now we hand over the session to Professor Arunachalam Ramaiah. Now the session is yours, sir. Welcome you, sir.
First of all, I would like to thank the organizer, Dr. Bala and Dr. Joseph, for organizing this fantastic international conference, which will be benefited to the young students, researchers, and the faculty members. For the today talk, I'm going to focus on how we have performed the genomic surveillance of SARS-CoV-2 and how that can be benefited for the public health official to take the necessary action to mitigate the pandemic outbreak. So I'm from the City of Milwaukee Health Department, and you can see the background and where our beautiful city and our health department is located on the right corner. Next, please. And you can see the closer view of our health department, and which is just located right next to the city hall of the Milwaukee government. Next. So already Dr. Vaiti and other provided some of the enough introduction about the SARS-CoV-2. However, I would like to cover some of the uncovered topics here. So the SARS-CoV-2, this is a severe acute respiratory syndrome, coronavirus 2. This is belongs to the Coronaviridae family. So initially that this virus has been identified in human in 1960, and also this virus is actually widespread, which infected a lot of other animals as well. So as of now, that human being were infected with 6 different types of coronaviruses. And recently, there are 2 types, like the SARS-CoV-2 1 in 20 years back, it's actually caused the outbreak in China. And in 10 years before, there is another virus called there's a MERS-CoV, this Middle East respiratory syndrome coronavirus, which caused the outbreak in the Middle East. So the initial outbreak happened in Saudi Arabia. Then in later 2019, the new virus of SARS is caused the outbreak or initially identified in the Wuhan city in China, and is slowly started spreading to the other countries in the continent. Is when we started, Dr. Vaiti and I started working on this virus. Only there's a few countries like China, Thailand, Japan. Those countries have reported few cases, and also that we are about to complete the project. At that time, that the US has reported almost 8 cases. So we have used those data to analyze and study the evolution of this virus in the earliest stage of this outbreak. And as of now, this virus is spread globally, and you can see the color coding in the world map. There's a large number of infected cases have been reported in many countries. So particularly in the American continent and a lot of Asian countries and the Europe as well. And as of 23 January 2023, that there are 664 million people worldwide infected with this SARS-CoV-2 virus. And also 6.7 million people have died because of this COVID-19 outbreak. And you can see the bottom table. You can see that even though the US has a less population, but there are 100 million people have infected with this virus, and almost or more than 1 million people have died because of this outbreak, which is comparatively less when compared the data with the Indian data. Next, please. So when US have identified the few cases, and the CDC started immediately working on this public health issues, and they got the specimen, and they pictured they took the 10 images of the specimen, and you can see that a lot of the blue particles, those are the SARS-CoV-2 virus. And they made the CDC made the cartoon, and it's located on the right side. So that's giving the clear picture how the coronavirus SARS-CoV-2 is looks like. And the red spikes, those are the spike protein, which is necessary for this protein to bind to the ACE receptor of the human cell. So that's how it's interact with the human cell for the infection. Next. And we have the generation of sequencing technology to sequence the ribo and the deoxyribonucleic acids. And so very initially that people have developed the sequencing technology called Sanger. That's the first generation sequencing technology. Even the Human Genome Project, they started with this kind of sequencing machine. And later, the second generation sequencing technology has implemented, which is a short read sequencing technology. That's there are multiple companies on the market. Like the named is Illumina 454 or Eintoran. Those are the second generation sequencing technology. And the third one, so currently many people are currently using this kind of technology right now. And this is a long read technology, and that can cover, and most of the people using this technology to sequence the eukaryotic genome, because it's very hard to sequence the chromosome level genomes. So with all this technology, so currently that many public health laboratories and the other research scientists, they are mainly using the Illumina as well as the long read third generation sequencing technology to sequence a lot of the viruses, prokaryotic organism as well as the eukaryotic organism. Next, please. And here is a picture. You can see that the length of this SARS-CoV-2 genome. So this SARS-CoV-2 genome has the 30 megabases or 30,000 base pairs. And this majorly, this genome is consists of the polyprotein called ORF1AB. Also, it has the four structural protein called spike, envelope, membrane, and nucleocapsid. Apart from all this protein, it is also has the five accessory protein that are required for this virus for successful replication and the assembly in the infected cells. So here, even though it has multiple proteins, the spike protein is a very important protein, as this protein has a domain called receptor binding domain, which can be used to bind with the human cell receptor. So through that interaction, this virus is actually entering into the cell for further replication and the infection. And you can see the well map provided here, created by my former student Drew, and where he beautifully plotted the number of the proportion of the cases, the proportion of the SARS-CoV-2 sequences submitted to the GSI databases. As you can see that the more sequences have been submitted by the United States and Canada, and followed by the European countries, India and Australia. And there are only a tiny proportion of the sequences have been submitted from the African continent. Next, please. So, people have many laboratory from the worldwide started sequencing the SARS-CoV-2 genome. And what is the necessary of having the genome? That's the first question we need to ask. So the entire purpose of having the SARS-CoV-2 genome is to identify the circulating variants, and also it will help you to identify the transmission pattern of this virus. So based on this information, the public health official can take the necessary action. So for here, when that US has reported few cases in early 2020, and we have started working on that virus sequences. So the phylogeny provided on the left side shows that so the the clade with the orange background is a SARS-CoV-2 genome or the strains, and you can see that the yellow one shows the first SARS-CoV-2 one viruses, and the bottom one shows is a MERS viruses. So this phylogenetic tree is beautifully divided into the two clade, and the one is completely formed with the SARS-CoV-2, and that actually closely related to the bat viruses. So it shows that the currently circulating SARS-CoV-2 virus shares almost 96 sequence identity with the virus that isolated from the bat. So, which means that the virus is possibly evolved from the bat viruses. And also we have done the comparison with the previously isolated bat viruses in 2017, as well as the SARS-CoV-2 one virus as well. But the 2017 bat virus shows only 88% sequence similarity, while the SARS-CoV-2 one as well as SARS-CoV-2 shares 80% sequence similarity. That confirming that the SARS-CoV-2 it's not actually evolved from the SARS-CoV-1, but is actually evolved from the bat virus by acquiring the additional mutations as well as the insertion and deletions. Next, please. And you can see the how the variance is evolving. So when China has reported the first sequences of SARS-CoV-2 in the database, and on many laboratory from the different parts of the world, and started sequencing the infected cases. And they also subsequently submitted all the sequences to the database. And so after the few months, and people started seeing the lot of variation and the genetic mutation in the new viruses. And since the people started looking at a lot of variation, particularly on the spike protein, and they started classifying those new variant, the new mutation, or the strain which contain the new mutation as a new variant. So in that way, they have started classifying the strain as a alpha, beta, gamma, and right now we have the Omicron. And you can see the picture phylogeny provided on the right side. You can see the red and orange, those are the Omicron strain. And right now that it has almost 40 to 100 mutation when compared to the reference strains initially sequenced by the Chinese laboratory. And also we have estimated the evolutionary rate of this virus, and it shows that this virus actually gaining almost 30 mutation per year. This is something we are expected that the RNA viruses evolve faster than the DNA virus, as well as the bacteria and the higher eukaryotic organism, because the DNA virus, bacteria, and higher eukaryotic organism has the much higher genome size. So this RNA virus has only the 30,000 base pairs, which is 30 megabases in size. But if you take any bacteria, for instance, if you take the Mycobacterium tuberculosis, the causative agents of the tuberculosis, so the genome of this pathogen is only the 4 megabases. In the case of the eukaryotic, if you take the Anopheles stevensii, this is a mosquito which can transmit the malarial parasite. So the genome size is 250 megabases in size. If you take the human genome, this is more than 3 GB in size. So compared to all this organism, the RNA virus, particularly the SARS-CoV, has a less genome size, but is highly, the pathogenic virus, and which has a higher rate of transmission right now. Next, please. And so when right now the database, a specific database called the GISAID database, that has the all the SARS-CoV-2 genome information. And my student Guru have pulled out all the data. This is when he was analyzing the data a couple of months before. The database contains 12 million sequences, 12 million SARS-CoV-2 sequences, and we pulled out the sequences for each of 5 variants, and we made the when to compare. That is, what are the mutation or actually commonly shared by the 5 different variants of concern, and how many number of mutations are unique to each of these variants. That's located on the top left of this picture. And also, since the spike protein is very important for the public health action, as well as the studying the lot of basic biology related questions, we have pulled out all the spike protein from the 5 variants of concern, and we have identified almost 1,741 unique mutation, or the common mutations shared by all the 5 variants of concern. And on the right side, you can see the modeled structure of the spike protein. And this is a three-dimensional structure, and where we have highlighted the top 10 the mutations. So you can see that I have listed the mutation and the frequency of that mutation in the 12 million sequences that we have studied. So the overall, the mutation is actually present from 50% of the strain to 99.5% of the 12 million sequences deposited in the database. Next. And this slide shows that how the SARS-CoV-2 variants actually evolved in a different timeline. So you can see the first plot that you can see the x-axis, which has the timeline starting from the January 2022, January 2023 right now. And you can see it shows a very clear pattern that most of these variants of concern that actually evolved in the late 2020 as well as late 2021, and almost in the November of 2021, that the Omicron strain is started evolving, and is right now there's a most of 99.9% of the circulating variants are Omicrons. And this is a very broad classification, and people who's working on the surveillance project, and they dig into see that what are the specific the lineages or sublineages or circulating, because those are the identifying the circulating variants are necessary for our public health action. You can see the picture on the right bottom, and you can see that the number of sequences have been submitted to the GISAID databases. And you can see that the most of the sequences have been submitted from October 2020 to the mid of 2022, and then it started declining. So now, from worldwide, there's only that very less number of sequences being submitted to the GISAID databases. As we are finding there's a less number of cases throughout the world. And you can see that the comparison table located on the left bottom. So as I mentioned, we have right now that 664 million cases reported, but you can see that only the 2.2% of the infected cases have been sequenced, and the sequences have been submitted to this GISAID database. So the US has reported the maximum amount of the SARS-CoV-2 sequences, and almost more than 30% of the sequences submitted in the GISAID databases are from the United States. And in India, we have just submitted only 0.6% of the infected cases sequences in the databases. Next, please. And as I mentioned that in the previous slide, I have showed the broad classification of the variants of different variants of concern. And here we have provided in-depth detail of the circulating lineages as well as the sublineages. You can see the plot on the right side that shows that's based on the last 3-month data. That shows that the currently circulating or dominant strains are XBB 1.5 as well as the BQ1 and BQ1.1. So almost 90% of the circulating strains in the United States belongs to these three lineages. Next, please. And here the CDC, there's a health department, human health and services, have grouped the 50 states of the 50 states in the US into the 10 different regions. And you can see that the picture on the right side. So as you can see that most of the East Coast states or East Coast regions are dominantly infected by the XBB 1.5, but is completely different than that the what actually the other regions have reported. For instance, the Mid East here in the 5, as well as the mid US, as well as the West Coast, that's California, so it's all reported the BQ11 as a dominant strain, and you can see the big difference in the pattern of circulating strain in the different US regions. Next, please. And so in our public health laboratory, as of now, we have sequenced almost 10,000 SARS-CoV-2 genomes. And so those specimens we have collected from the multiple sources, like the samples came from the local community, school students, universities, populations, from the jail and shelters, and so on. And we analyze all the samples as a one group, but for the research purpose, we segregated the university population sequences and done the in-depth analysis on that. Next, please. So here, when we collected the almost 1,717 samples from September 2020 to April 2022, then we started sequencing that samples. So you can see that on the plot on the left side that in the first 3 months we were setting up the facility, which means that we are setting up the sequencing machine, and we are trying with a couple of the sample to see the efficiency of that sequencing. So that could be the reason we couldn't able to sequence much of the submitted specimens in the early stage. And you can see the sequence of the specimens are colored in the gray. And from the December 2022, right now, we are successfully nearly sequencing almost all the samples that we are receiving from the different community. So in this study, from the December 2020 to the April 2022, we have received 771 samples. Almost 70% of the samples we have successfully sequenced. So then after sequencing 562 SARS-CoV-2 samples, after removing the low quality sequences, which mean that any sequences identified with the less than 90% of the genome coverage, as well as the depth is, if it is less than 100%, then we can remove that the sequences. So finally, we have considered 537 sequences. You can see that the circulating pattern on the graph located on the left side, and it has a multiple colors. And you can see that the currently circulating Omicron is actually we have started reporting or started finding that Omicron in the university population from the November 2021. And dominantly, we identified the sequences from the Omicron and the Delta clades. Next, please. Nate, next. From the two university population, we have successfully sequenced 537 sequences. Apart from identifying the broad clade, we also identified the lineage as well as the sublineages as well. So here we identified the dominant lineages as a BA 1.1 as well as a BA 2.12.1 as a dominant lineages in the university population during the time period when the samples were collected. So this is in line with our finding that US has reported almost 48% of the sequences submitted in the GISAID databases are actually from the United States. And also 81% of the sequences available in the database are submitted by the United States. Those sequences are belongs to the BA2 12.1. And this is the reason behind that probably that why that US has identified these two subvariants predominantly. This is possibly due to their tight or the strong replicative fitness, immune evasion, and also the resistance to the monoclonal antibodies. Next. We also compared our findings and the lineages information with the local community, as well as the data from the Wisconsin state, because we are from the Wisconsin state, and so we compared our data with the Wisconsin data as well as the national data. So you can see that is all the data shows a similar pattern, even though the number of sequences submitted from each region is varied, but the pattern is still the same. For instance, you can see that the Omicron, that's a GRA, that's a purple color, is started, all the plot is started showing from the November to or the December 2021 to right now. I mean the till date, that's most of the 99.9% of the variants are belongs to the Omicron. Next, please. This is a phylogenetic maximum of likelihood phylogenetic tree we have constructed for this 537 sequences. And this tree is actually has many clusters, and the main clusters are the Omicron that are highlighted in the yellow color and the orange on the top. And the bottom, it shows that the delta strains are in the green color. And so the branching of this tree is completely based on the mutational differences in the sequences that we have studied related to the reference strain that's initially reported by the Chinese laboratory. Next, please. Apart from identifying the phylogenetic relationship of all the 537 sequences, we also constructed the phylogenetic tree for these sequences along with the global sub data set sequences. So as I mentioned, right now we have almost 14 million sequences in the database, but it's very hard to handle 14 million sequences for the phylogeny, considering the computational requirements. So we have subsampled the 14 million, and we have obtained almost 2,500 sequences. And we combined the 2,500 sequences with our sequences to see that whether the university strains are closely clustered, or is it completely is a kind of combination of strains which has closely clustered with the sequences from the other part of the world. Next, please. So this is a in-depth look or the zoomed-in version of that phylogenetic tree. You can see that the strains from the other parts of the world, or the other community, are highlighted in the green dot, and the university sequences are tightly clustered together, indicating that they are very closely related, and most of that population are infected by the genetically very similar viruses. And you can see that the number of mutation present in the Omicron, we have identified is the number of mutation in the Omicron is more than 40. Next. And this is one of the important slides. And here we have identified the transmission pattern of this virus in the university population. Even though we have identified the different lineages of this virus, like including the Delta, Beta, and Omicron, for this analysis, we have focused only on the Omicron strains. And very interestingly, we have identified that the certain Omicron strains are circulating within the university population, as well as between the two university population. And also we have identified some of the strains successfully transmitted to the local community, and also vice versa. Also, we have interestingly identified some of the strains in the university population. They didn't transfer to the other population, but is disappeared somehow. And finally, that as expected, only the few Omicron strains successfully transmitted within the university, or between the university population as well as the community. And also, when we say that the particular strains is transmitted from the university to the community, or between the university population, we also done the Pyrois comparison, and that also confirmed that the strains or just the maximum it has the one mutation in it. Next, please. It's a very close view of the phylogenetic tree, and you can see that the particular clade has the sequences from the two universities. And here you can, this is another kind of evidence that shows that the transmission occurred within the university as well as between the university populations. And not all the strains, but some of the specific Omicron strains that actually successfully transmitted between these groups. Next, please. So overall, that this study is overall studies conclude that or emphasize the importance of this SARS-CoV-2 genomic surveillance, and this is not only just important to characterize the virus, but also is important to identify the circulating variants and the transmission pattern. Those are important for the public health official like us to make necessary public health action further. Next, please. So I'm almost at the end of this presentation, and so the most of the public health laboratory they do the sequencing. That the process is known as a genomic surveillance of different the pathogens, even including the monkeypox viruses as well. And while we take enough effort to contain the diseases, I strongly believe that it's our individual responsibility to maintain the good health. So the good health has the physical health as well as the mental health as well. And for the physical, maintaining the good physical health, and everyone should maintain these good sanitary procedures, and also the doing exercise, having the proper nutrition, as well as getting the vaccination. So those are the four key points are necessary. And also finally, that maintaining the personal hygiene is utmost important. So doing all these things that help you to stop spreading the many infectious diseases. So with this, next slide. With this, I would like to thank the team. And the picture on the left side is my current laboratory, and many people have involved in this project, and most of them have received the samples and done the sequencing. And myself and my team were analyzing the entire data, and also the former student Drew, and the long-term collaborator Dr. Vaiti from UCLA. You can see the picture on the right side. When I visited his laboratory in 2014, we took this picture, and the middle you can see Dr. Joseph as well, and we all were very thin, almost 9 years before. And so without their help, it's very hard to do all this work. And I finally thank my funders, CDC and doctors, for this wonderful work. And thank you everyone for this great opportunity. Yeah, stop please.
Thank you very much, sir, for your valuable insights on the molecular basis of various strains of COVID-19 viruses which is available all over the world. Thank you very much, sir, for your valuable speech.